Okay, so an enzyme has an active site, which is going to do one of those four things I talked about in the last lecture. That is somehow going to be able to interact with a substrate to produce a product. What we now know happens is once the substrate or substrates are in the active site, we see that the rest of the enzyme will change its shape. Change its shape in the process of allowing the active site to cut, paste, twist, tweak, whatever. They call this the induced fit. You look at the figure right here. You'll notice here in the first, the active site kind of sort of has almost the shape of the substrate. This curve and then the triangle. As the enzyme's active site starts to interact with the substrate, it will change the shape of the enzyme. And as it's changing the shape, what it's allowing to happen is the cut, the paste, the twist, the tweak, whatever. Here in this figure from the textbook, you see that a substrate, which is a polymer of two monomers, enters in the active site. The enzyme changes its shape, the induced fit. In the process of this induced fit, that polymer is cut into two monomers. Now, the specificity for the active site is such that many biologists, cell biologists, refer to this as a lock and key model. Think about it. Start up your car, you have to have the specific key that was given to you. No other key will start that car unless it has the correct shape. That's what all the little teeth, the jagged edges are. The correct shape is the only thing that enters into that lock. So the induced fit around the active site is referred to as the lock and key model. You can see here in this computer topography showing that the shape of the enzyme will change when the substrates enter into the active site. The shape, the enzyme will fold around, bringing more of the active site into play. Now, as I said in the last lecture, the active site is going to interact with the enzyme. It's going to, orient. It's going to give a specific microenvironment, more acidic, less acidic, with water, without water, whatever. And it's all going to be due to the fact that the active site, which is of the enzyme, and remember the enzyme is composed of polypeptides, the active site will have specific amino acids. Specific amino acids that have the R groups, as we see right here. The R groups are going to give that active site its microenvironment. The acidic amino acids are going to make the active site more acidic than the environment the enzyme's in. If they're hydrophobic, well, they're going to expel water from the active site. The R groups are going to allow for the enzyme to physically interact with the substrate. Well, how can we extraneously affect the ability of an enzyme to function? First one, suboptimal temperatures. Okay. Keep in mind, everything in the universe vibrates. They call that Brownian movement. The Brownian movement allows for things in a liquid to move. You lower the temperature, Brownian movement slows down. Things are slowly moving means that the substrates entering into the active site of an enzyme will be slowed down, which means the activity of the enzyme will be slowed down. You could speed up the process just a wee bit by adding just a little bit more temperature, more things vibrating. Problem is, you go a little too far, well, you cook an egg. That's basically what's happening. You raise the temperature too high, you do what they refer to as denature the proteins. Remember, tertiary quaternary structures held together by hydrogen bonds, disulfide bonds, ionic bonds, all that type of stuff. You start to add energy, you start to break bonds. You break enough bonds, polypeptides unwind. Polypeptides lose tertiary, lose secondary, and all you have left is the primary structure. It goes back to that thing I just alluded to, cook an egg. You add heat to an egg, it turns white. Why? You're denaturing the proteins due to thermal energy. You can do the same with lowering the pH, make it more acidic. All those hydrogen bonds holding the tertiary structure together, well, you now interfere with them because you now have extra hydrogens floating around. Because remember, you have a low pH, you have an acidic environment. Things will fall apart. In the cell, you can only do number 2. Okay? You can only use number 2 as a mechanism for control. You can change the acidity, the pH up and down within a vacuole, within an organelle. Okay? So you can change the ability of enzymes to function. Yes, no, depending on pH. You cannot change the temperature within a cell. Okay, there is no thermostat. 98.6 degrees Fahrenheit, 37 degrees Celsius. That's it.